In this mini lecture, we will briefly go over the data science lifecycle. What is the data science lifecycle? The data lifecycle, outlined here on the left, has 7 steps, and you're going to see if you search in the internet that there are various versions of this cycle, typically ranging from 5 to 8 steps. But basically, this outlines the process of analyzing data. These concepts originally emerged in the business world. As shown in the image, you can see the first thing says business understand in the first step. And they are often tailored toward business-related problems. However, the same approach and steps are widely applicable across different fields and types of analysis. For example, in business, many analyses involve collecting large number of data to develop some predictive model that identify trends and key features within the data. And we'll see that through data visualization. In a research lab, we not only collect large amount of data and make predictions from it, but we also conduct primary analysis, such as running experiments, testing hypotheses, and generating data like DNA sequencing results. From there, we move into analysis phase, where we interpret the data and share the findings with the world. When we think about this process, you'll notice that the same fundamental steps apply, even in this type of scientific data analysis. With that in mind, I want to introduce a more general version of these steps, listed on the right here, one that isn't limited to the business world. You'll come across many different labels for these steps online, but they all follow the same general structure. The process begins with defining the problem, identifying the key question we need to answer. Next, we gather or produce the necessary data, whether by collecting existing data or generating new data through experiments. After that, we will clean the data, a crucial step that we'll discuss later on. We will conduct some exploratory analysis to examine what we have and deciding how best to proceed with the analysis. We will select the relevant pieces of data to include in our study and move forward with the analysis itself. Once the analysis is completed, we move to the visualization and interpretation stage, making sense of the results and drawing conclusions. Finally, once everything is interpreted and ready for dissemination, we take an extra step. You see step number 8 is not in the chart. This extra step is for sharing our findings in some form, whether through a publication, presentation, or other means. This step is an additional step that is critical in the scientific field. Now, I'm going to go briefly over each of the steps. The first one, defining the problem, is arguably the most critical step in the entire process. Without it, there is really no reason to move forward at all. While it's possible to quickly come up with a problem statement, taking the time to think carefully and deliberatively at this stage is essential. This is where many people encounter issues. If the problem is too broad from the start, you may end up doing unnecessary work later in the analysis process, which could have been avoided by refining your question at the outset. A well-defined problem helps streamline the entire workflow, preventing inefficiencies downstream. On the other hand, if the question isn't framed with the right caveats or consideration, you might introduce biases that could affect the validity of your entire analysis. As you can see, this step is crucial in scientific research, particularly in biological laboratories. At this point, the hypothesis may already be well defined, but in other contexts, the hypothesis may come later, after formulating the initial scientific question. Finally, as you define your problem, you must also consider what type of data will be necessary to answer it. This naturally leads into the steps of gathering and producing data. In the business world, companies are constantly gathering and analyzing data. They maintain extensive database tracking customer financial trends and other key metrics. Much of their work involves mining these data sets and to extract insights that help refine their business models and improve decision making. In science, we often follow a similar approach. We start by looking for existing data, examining previous scientific studies to determine whether relevant data is already available to answer our research questions. However, a key difference is that in science, we frequently generate our own data. Many of you will likely do this throughout your research careers, producing various types of data, such as DNA sequencing data, bioimaging data, clinical data. The range of possible data types is vast, depending on the field and research focus. At this stage of the process, a lot is happening, and it can be a little bit overwhelming. One common pitfall is failing to properly document critical details about the data that as it being gathered or generated. However, it is crucial to take the time to record all necessary description and metadata in an organized and systematic manner. Doing so ensures that valuable information isn't lost and that your data remains useful and interpretable throughout the research process. Now that you have your data, the next step is cleaning it. Data cleaning takes many forms, but there are several key tasks that consistently need to be addressed at this stage. If you're working with data from multiple sources, you'll need to integrate into a consistent format. This often means ensuring that datasets using the same structure, such as aligning column names in a spreadsheet or resolving inconsistencies in labeling. These inconsistencies could be minor, like capitalization differences, or could be more significant. You have different terms referring to the same concept. Identifying and standardizing this discrepancy is a crucial part of the cleaning process. Another major challenge is handling missing data. You have to assess whether the missing values are minimal and manageable, or if the quality is so poor that it's unreliable and should be discarded. Similarly, with under-annotated data, cases where a data set may exist but lacks key details necessary for meaningful analysis. Understanding what data is too incomplete or unreliable to use is just as important as deciding how to clean and structure what remains. Ultimately, data cleaning is about ensuring that the information you work with is accurate, consistent, and useful, setting the foundation for meaningful analysis. Step 4 involves exploring the data before conducting any major analysis. At this stage, you're examining the dataset you've collected, performing basic analysis, and generated initial visualizations. This process is often called exploratory data analysis, or EDA. EDA helps with distinguishing between useful and irrelevant data and refine your focus. If you haven't yet formulated a hypothesis, this step is critical for defining one to test in the next stages. Following EDA, you move on to narrowing your dataset. This means removing irrelevant, noisy, or inconsistent data. Some advanced methods like deep learning can automatically detect and account for noisy data, but many traditional analyses require manual refinement. At this point, it's also valuable to compare your data against a gold standard dataset to ensure that your analysis produces expected and reliable outcomes. Once you refine your dataset, you move into the analysis phase. If you're working with predictive model, this is where you train, select, validate, and apply models to your data. If your analysis is not predictive, this step includes running statistical tests, computational analysis, or sequencing workflows, depending on the nature of your research. This is where the core analytical work takes place. Up until now, much of the work has been computational, but now you must translate raw results into meaningful insight that can be interpreted by humans. So this is how visualization come into place. This is a critical component. Choosing the right charts, graph, or figures to make trends and patterns in the data easier to understand. Beyond visualization, you must contextualize your results by comparing them to existing knowledge, allowing you to interpret findings in a meaningful way. Finally, an additional step that is sometimes overlooked but is crucial is the reporting and sharing of your results. Once the analysis is complete, it must be communicated effectively. Ideally, your findings should be stored in a database or repository, ensuring that future researchers or analysts can access and build upon your work. In industry settings, this might mean an internal company database. In the scientific research, results are often deposited in global repositories or published in academic journals, making them data available for broader dissemination and future discovery. So I hope you now have an idea of what the data science lifecycle entails, and we're going to see examples of some of these steps in the next few lectures. Thank you.